Class, this is Miss Wint, and I'm making this video to help you get started with the cell types gizmo. That is a simulation on the website explorelearning.com. And this website, you do need a username and password. It is probably the same as what you use for Schoology. We set them up the first couple weeks of the year. If you don't think you have a username or password, please send me a message and I will get you hooked up with one. Okay. So the paper looks like this. I've had paper copies in my room, but if you're doing it on Notability, that's fine too. And some of these vocabulary words we don't know yet, so I don't want you to stress out about that. A lot of them are explained in the assignment. And you have a couple prior knowledge questions that we do before the activity. Okay, so to kind of review, how do you know if something's alive? So we talked about, we had this activity with the Martian in the car earlier this year, and you had a packet with, we had like 9 characteristics that all living things had. All right, so what are some of those? So you need to kind of recap your memory, maybe go back in your notes. What are some things that you know about for living things? Okay, moving is not one of them. Not all living things move that much, like a tree. Okay, but what do they do? What parts do they have to have if they are alive? Okay, and then question 2, humans, plants, mushrooms, we're all alive. What do those things have in common? Okay, so question 1 and 2 are kind of similar to each other. You may have some similar answers, and that's okay. So you should answer these guys first. Go ahead and pause the video if you need to and get those answered, or you can go back later when you start the assignment if you're just watching this video all at first. Okay, once you get that, we need to go to the website explorelearning.com and get logged in. Okay, we're doing something called cell types gizmo. We're going to use a microscope to compare things. Okay, and we need to find something called the LOD leaf to look at. So let's go to the website and let's see if we can kind of get that far, right? So here's the website, explorelearning.com, and I'm going to hit this button up here that says log in. I'm not signing up for free. We have accounts. Mine pops right in there. You may have to type yours out. Try what you use in Schoology. Again, if you can't get in, let me know, and I will hook you up with the login. Okay, so username, password, hit login. You're good to go. I have a teacher screen before you. You don't have this screen, but yours will look like this. And so the current assignment here, we have a cell types. And I'm going to launch that. I'm going to turn that simulation on so I can do my activity. It takes a minute to kind of think about it. Now my stuff up here is like teacher things, so you don't necessarily have all those things. But what's in the box here, this is what's important. Okay, so we have a picture of a girl. You see if I move my mouse, some things get highlighted. Right, and there's a box here that says show all samples. Okay, so this is all the stuff I can look at under the microscope. Okay, so I need to pick something, and my question said I needed Elodia. So that's right here. That's this plant, this leaf. So I'm going to click on that, and then it lets me click on microscope because I've chosen something. Okay, so here's my microscope. And my directions say drag the coarse focus slider until the focus sample is as focused as well as possible. Okay, so we always start magnification on the lowest setting, and we always start with the big knob, the coarse focus. And you kind of drag it back and forth. You can see as I move this, the stage on my microscope is moving up and down as well. Okay, so this is moving that stage, and it makes things come in and out of focus. So pick a spot where you think it looks pretty focused for your eyes, and whatever your eyes is may not match your neighbor's eyes, and that's okay. Okay, and then improve the focus with the fine focus slider. So here's the fine one. So this changes it just a little bit back and forth, and I want to make it so I can kind of see those little dots inside. So maybe right there or so. And notice when I move this one, I can't really see the stage move. It's moving such a small amount that I can't really see it with my eyes, right? Okay, so this big knob was the coarse focus. The little knob is the fine focus. Okay, so number one says, what do you see? Well, if I look here, I see kind of rows of green something. They have a green outside and kind of an empty inside. Kind of like a brick wall. So if I go back to my worksheet, what do I see? I see a green, I'm going to say brick wall of boxes. Okay, you can put it in however words works for you. That's what I'm going to go with for my answer. Okay, so I did all this stuff here. I grabbed the leaf, put it on the microscope. I changed the focus. I used the fine focus to make it even better, and that's what I see. Okay, question 2. Now we're going to change the magnification. This 400 is a bigger number. And it says, if necessary, adjust the fine focus. It doesn't say to touch the coarse focus. And the reason for that is, I'll show you on our simulation here. If we jump it up to 400, we zoom in really far, okay? But the lens here, you see as it kind of goes through the different lenses, this one is the longest. And if I move that coarse focus, this stage going up and down may crash into the lens and break something. So we only use the fine focus with the higher powers. Coarse focus I can only use on the lowest power. And I'm not even sure if it'll let me. Oh, it does let me. But you should never use coarse focus unless you're on the lowest, shortest power. I can use the fine focus to try to make it as perfect as possible. Okay. Now these buttons here, you can see I kind of can move things up and down if I need to draw a picture or get something better. Okay. So now if I go back to my paper here, now what do I see? I can say boxes got larger, and inside there are, and then you can fill in the blank. What do you see inside those boxes? Okay, so inside there, so there's my box. I can see there's little green circle things floating around, right? And there's a big empty space in the middle. So those are things you can describe on your paper. Okay. So that kind of shows you how to use the microscope. There's another button here that says test for life. We are not doing that part of the experiment today. So don't worry about it. I mean, you can mess with it, but there's no questions with that. Okay. I can also show labels of things. Oh, hey, there's some cell parts that you maybe or maybe don't know yet. And I can also show how big something is. So that's a micrometer, 20 micrometers across. Okay, if I change it to 100, change it to 40, you can see how that's zooming in and zooming out. And you can see here the different lenses, these are called objectives on your microscope, give us those different viewpoints by spinning them around. Okay. So it says the individual chambers we see are cells, the smallest unit of an organism. Well, that's cool. So each little green box is a cell. All right. So as you go through this, you are going to be looking at some different things from that environment under the microscope. Okay. So you have a little spot here where you need to match up microscope parts. Okay. So do the best you can with the description. So you have a large knob and a small knob. Knobs are circles usually. So that's probably C and D, right? Large and small. And just work your way through, pick the best stuff. If you have to Google a picture of a microscope to get that, that's fine. You are going to pick a sample under 40 and then change it to 400 and focus it. What's easier to use? What things can you see in human skin? What things can you see in other samples? So you're going to go through this step by step and get as far as you can. Different types of cells and what can you see inside. You have to estimate the size, so you have to turn that ruler on to be able to see the size of things. Okay, do a little measuring. How big is this thing we're looking at? Okay, so that's what this paper is all about. Okay, is using that microscope and trying to look at all the different samples of things. Okay, so when you're ready to change samples, I want to get rid of that LOD leaf. I go back to the landscape, and I can click on any of these lit up things. Okay, so like a neuron, that's a brain cell. That might be kind of cool. Put it under the microscope. Use the coarse focus till I can see something. Right about there. And then make sure it's as clear as I can get it. And then change it to 100. And a 400. And I can see all the little parts in there of a brain cell called a neuron. Okay, so you're just going to be checking out some different things and practicing focusing in prep for when you actually get to use the microscopes, and also learning a little bit about the parts of cells and seeing what all these living things have in common and what's the same and what's different. So that's what this assignment's all about. If you have any questions, please let me know. Otherwise, happy hunting, have fun with it, work with a partner if you want to, and we'll talk to you later.